Analyst must know HPTLC.
Thank you, Mr. Chairman. I would like also to thank you, the organizing committee, to invite me for this talk, especially Syed and David. Okay, so I will try to tell you some words about HPTLC. It's a little bit as HPLC, but there is a T in addition. So the first thing, how it started. It started very in Russia in the 30s with so-called ultrachomatograms. And at that time, the goal was to check the drugs and to avoid any problem. So they make only on small slices of microscope, and they make aluminum oxide, for instance, and they make these nice spots, which were called ultrachromatogram. Then it goes to the US already, and with Kirchner, who invented the TLC, as you know, at the moment, because he had problem with the column which were not stable to analyze the juice of orange or grapefruit, and that was the start of the TLC as we know at the moment. It came back to Germany in the 60s with Stahl, who invented the TLC, with Merck with the TLC on silica. And then the high-performance TLC, which gave this name with HPTLC, starts in the 70s, so it's already an old story. And a great breakthrough was made by Berger. It was for working from Bayer. I will not explain all the story, but he makes a very nice thing with a gradient. AMD is automated multi-development. It was a gradient. And you see the separation, and this is made on the TLC plates. And also what is interesting is that you see that you detect already at low wavelengths, which avoid any problem with the solvent. Then duochrome, it was a coupling already with TLC and HPLC, and then lycrosphere, the first phase, spherical phase. And finally, the monolithic phase in the year 2000 around. And in the Pitcon 2005 DART, which wins the Pitcon Golden Award. This is why, you know, the TLC now starts again. Another milestones was in 2007. Usually it was nothing about TLC, but in Ghent, I give a call to my friend Pat Sandra, and I told him, well, if we place an abstract, would you have a lecture? And then my friend Gerda Moloch made a main lecture. Then the year after, she made a very nice lecture about HPTLC and showing the speed of the method, which is very interesting. And then we have a TLC interface made by Camag, a TLC interface with MS. It was the start of the real renewal of the TLC. Then we had a symposium in Basel. We went up to 330 attendees in comparison with Interlaken, which was the previous event in the late 90s, and we have only 70 participants. So it's a real trend at the moment. And in Berlin it was this year, and we will see the next steps. The first thing I would like to point out that people usually think about TLC as this way. You know, it's in a small part of the lab. It's a very old method, and nobody wants to do it. But it is still a liquid chromatographic method. And already in the 80s, you know, the people have statements that shows that it is a problem that the people have no knowledge and they do it, with feeling and with this not giving very good results. Even with a specialist in the field, they don't know. For instance, if you ask to your colleague in the analytics, very few people know about the gradient. And once you say about other features of the method, they don't know that there is a gradient in TLC. They don't know the results that we can achieve with this, and also with the coupling possibility. And especially, is it quantitative? You know, of course it is quantitative. You see, this is a validated method on stereoside in different thing, and we got very good results, and it was validated. It was shown in 2013. This, and the interest is that we have very good results in this concentration, and especially in heavy matrix samples. And is it sensitive? Yes, it is sensitive, and it's reliable because we check also with the sample and with the reference together. We say that it is. We see that it is the same product, and we can go down to a very low concentration. So it is safe and immediate confirmation, and it's also sensitive because we apply more volume. on plate you see what you need. So this is what is interesting, especially with dyes, but not only. We have many things. And also it is quick, you know. If you try to compare with other methods, I'm sure that you will be, it will be very difficult for you to achieve this, you know. Only 20 seconds per sample and only 200 microliter of solvent. The trick is that usually it is known for identification. So, but why stopping here? You can do everything quantitative and also mass coupling with this interface. So now, usually the people are making manually TLC. And once you see manually, here it's a protein dimers that the people will check about the lipids, the parietal lipids of this protein, of this dimer of proteins, which is extracts from a bacteria, a marine bacteria, Rhodobacter blasticus. And they do this in hands, you know, by hand, you know. You can get any result with this way, you know. And we do exactly the same sample, same concentration, and we analyze even the minor species that we don't see it there, you know. So it was very good achievement with just with a method, a proper method, proper instruments, proper plate, using this with an automated system. It works very well. And we make a paper because the aim was a crystallization of this protein, and we were quite successful. Also, what will interest some of you, it's to make clinical batches faster. You know, this is made by a coupling between the prep LC with big columns and also HPTLC. So you first, I don't show it this here, but you first set the method on TLC and you transfer directly on this column, and then back you check the fractions, and it's very efficient. It is safe. You have the real purity. These people, they purchase an intermediate. It was shown to be 98% on the UPLCMS, and they check it. It was actually only 70%. So it was 30% of impurities. So it was not a problem for them. They use their method, and they get the impurity pure, and they get their compound within half a day. So it's efficient. And of course, it's higher resolution before MS or NMR, because you can do MS and NMR on it. Of course, you can forget the whole TLC. That's no nonsense. And they have this instrument for the building with columns, big columns of 50 centimeters width, and they have the complete system for HPTLC. This name is known. Now there are 0 CMOs, so you can contact them. They make this also for you. This is another aspect. You know, sometimes we don't know what you are searching for. So with this multi-detection, it is interesting because you can check anything, you know, and you can do a Vibrio fischeri, so it's a bacteria which is sensitive to toxin. You can see also the acetylcholinesterase reaction on the plate, so you can do a lot of things. So it's interesting, and it's non-targeted. And afterwards, you can, of course, identify your compound by coupling with MS and NMR. This is something else, you know, just for your information. We have also very good developed software to speed up with PCAs. PCA sometimes is very heavy, and we have a proximal multivariate analysis, and in only 3 minutes you can achieve very good results. And also artificial neural networks helps to improve the noise ratio. It was published, and they win a Young Scientist Award this year in Berlin. A lot of people speak about endocrine disruptors. We go down to the femtogram per zone, so we have also a very interesting method, and you can see the film on the net. Back to the peptides, because it is the topic of this symposium. We have plates by Merck, you know, where you can do this analysis of peptides. You have many possibilities. This is commercially available. And I would like to show you something that has been made already maybe 15 years ago by Ibsen, and it was a quantification of amino acid in peptides. We were successful. They have a method which was not readable with a big automate from Hitachi, and it was not working that well. They have a lot of problems. And with this, it gives a good alternative, and it was transferred afterwards in the plant in Ireland. For cleanup validation, it's also an old story. It was by Lonza, and you see here the range of quantification, and here before cleanup and after cleanup. And you have here the sample after before cleanup, and then after cleanup. So it was a very good method also to check the cleanup validation when you are under process of manufacturing. And this is unpublished yet. It's something made by a cosmetic and pharmaceutical company. And the problem was that to analyze this in their matrix, which is a gel, a cream gel, it was nearly impossible with HPLC, because if they use SP coverage, they don't get reliable results. It was not sensitive enough and so on. So it is just simple. They put the staining media together with a solvent, and they make a short migration. So also it's very quick, you know, because it's only some minutes, 4 minutes for the migration, including the staining for the whole analysis, not only for one sample. And here you see the chromatogram, it's quite clean, and the calibration curve. And I think it's a good example that it is very interesting for speed, for reliability, and for cost. This is the reason why industry is interested in this. So now shortly the characteristic of the HPTLC. You have a many characteristic, high matrix tolerance, reduced cost. It's parallel, it's flexible, you know. But the main thing is the coupling and the bioactivity detection and the detection in general, which gives you a good selectivity for your analysis. And especially for coupling, why? Because with other methods, after separation, the sample goes to the waste. Here, after separation, the samples remain on the plate. So you can do a lot of things. You can do micro cells and a lot of things. This is an example we speak a bit with some colleagues. It's on sponges. You know, it was a problem to get the information about the toxins from sponges, and we go with this multi-technique coupling online, and we get then all the information about these toxins, especially for those toxins who are not you cannot detect easily. And why we detect more than with other methods? Because we dip this plate into a mixture of cells and a mixture of bacteria, marine bacteria, which will be sensitive to the toxins. And of course, we see the reaction of this on the plate. And on the same line, we can make an MS, MSMS, or NMR or FTIR, and then we identify this molecule very quickly. So it's a way to speed up the research with this multi-coupling. We can do a lot of things, as we see here, and of course, the trick is to make MS coupling. You can make MS coupling with elution on the plate, but we can do also desorption. And the elution is what we advise. It is a patent by Luftmann. Okay, and then I think I'm more or less in my time, okay? It's a quick method, you know, so it's no problem. And this is something that my colleague, a German colleague, which is a co-author of this talk, like to point out, because, you know, we are not in the mainstream, as you see. I think most of you don't hear already this kind of results with this method, but it's just an alternative method. So it's not to put away other methods, but to say you can do this as well, you know, and you can also confirm your results. And we don't speak, I don't point out in the previous talk, previous workshop, nobody speaks about uncertainty of the results, because you give very sharp figures, but we don't know plus minus what. And this is a way also to fix this a bit more. And just for fun, is it a bonsai forest? No, it is a plate where the people put seed to check anti-germination product. So the spot was applied there, it migrates this way, and you see clearly that there is anti-germination because then the grains doesn't show up. So it's a little bit of fun. So what miss mostly to do at TLC, it's to have open-minded and to have some ideas that you apply on the plate, and usually it works. And we are currently struggling to make a cell alive on the plate as well. So I think also for the chemist, we can do also synthesis on the plate and then migrate. So it's a lot of things going on. I think if you want to know more, we have websites, we have meetings. There is also in USA a company called CAMAG. It's located in North Carolina. You can contact them. So I think, and just for an end, you know, I was the last French talk of the day. So je vous remercie de votre attention. And this is also TLC. It's TLC if you type Chromart by Albert Alpap. TLC is also a way to make some art, you know, if somebody likes to. Thank you.